All right, welcome to our last day of data visualization. So I'm very happy to introduce Niels Gehlenborg, who is a professor at Harvard Medical School from a lab that does biomedical informatics, focusing on visualization of biomedical data. Thank you.
Thank you. Thank you very much for the introduction. So I understand this is the end of the course for you, and you've probably already learned a lot about data visualization. I apologize, the line was always one down there. And I want to maybe round this off for you with a topic that hopefully is relevant to some of you, and that's genomic data visualization, which is quite interesting from a visualization point of view. And I will try to explain to you why visualizing genomic data is challenging, and building tools for genomic data visualization is challenging, and what we can do about it. And I will then introduce you to a specific question, that is how to visualize 3D genome interactions in a web browser, and how to do this. So I think we have 90 minutes, almost 90 minutes. At this point, I'd be very happy for you to interrupt. If you have a question, I will treat this more like a lecture of sorts, so feel free to ask if something is not clear. I might also ask you a question to make sure you're still awake. All right, so visualization of genomic data. First of all, I want to show you that we have really enormous variety in genomic visualization approaches. Here are just a couple of examples of genomic data visualized in the sequence context. So this is one thing I want to emphasize. Everything I want to say today is about looking at genomic data in the context of the actual sequence. Because you could also, of course, measure gene expression levels, and in a way that's also genomic information, and you could create a heat map, or you could create an offset plot, which here you have learned about. That's also genomic data, but it's not visualized in the sequence context. So what's important here is we want to maintain the information that's contained in the sequence and the order and the structure of the genome in these visualizations. And I'm pretty sure you recognize a couple of these visualizations in here. Who recognizes one visualization? Who has maybe played around with any of these? Yeah. Do you recognize one? Okay. So which one? Anyone do you recognize? Is there one that stands out to you? The TAD plot. TAD plot. Okay. Here.
The top row, second from the right, is IGV genome browser.
Yeah, that's a genome browser. It's not AGV. It's a UCSC genome browser. Yeah. I think we have IGV on here as well. Anyone recognize things like this? So bacteria. I think this is actually a human genome. We have a bacterial genome visualization as well. So these round things, which are often either bacterial genomes or Circos plots. Okay, great. So you've seen some of those. The ones we'll talk about today in more detail are actually this one and this one. So you can look forward to learning more about those. Okay, Circos. Circos is a very popular approach to visualizing genomes. The idea of Circos is to draw a big circle, map the genomic sequence onto that circle. So along the outer edge, you have chromosomes listed here, and then you can map all sorts of data on the circle. You can map connections between different parts of the genome, whatever they mean here. You can map genomic regions, and so on and so forth. So that's a Circos visualization. You can look at something like the UCSC genome browser. I think everyone here is probably familiar with what a genome browser does. It is very similar in terms of the actual fundamental idea. Here the sequence is not circular, but it's linear. And you just map information to the positions on the genome. One challenge though with these things is often that the list of tracks that you want to add here gets pretty long pretty quickly, which then turns us a bit into a visualization problem, because now you can no longer see what's at the top of the page that we might want to refer to, or we might want to correlate a pattern we see here with something that's further up. So that's from a visualization point of view, it's always a problem to scroll around, because that means you can no longer see what your visualization was meant to show. Another example is the integrated genomics viewer. The integrated genomics viewer, in this example here, shows us a very detailed view of a particular region of the genome. And the reason why I'm showing you this is here you see essentially really individual nucleotides and SNPs, variants in sequencing reads that are mapped to the genome. And the interesting thing here is very clear when you see patterns like this, when you map sequencing reads, there's potentially a deletion here in a genome. In this case, they were able to confirm this by looking at some paired reads that are found on both sides of this deletion. Yeah. So I don't need you to understand any specifics here, but what I want to point out is when we go back and look at surface, we see a whole genome. Right? That's the whole genome. There's nothing missing really. We have the whole sequence of the genome shown. Of course, what's missing is detail. Right? This is an overview. Genome browsers, as you know, you can zoom in and out. In the case of the UCC genome browser, you can look at fairly large stretches of the genome, but then again, you lose detail. And in this extreme case here with IGV, you see, really the genome at the highest level of detail, and we're not quite down to seeing like the actual nucleotide sequence shown here, but we can, in this example, you can see this, we can zoom in far enough to actually see it. So this is also one of the reasons why there's so many visualization tools for genomic data, because we have to deal with very diverse scales, whole genome down to individual nucleotide. That is challenging from a visualization point of view, because obviously you cannot squeeze, in the case of human genomes, 3 billion nucleotides on a screen and then expect anyone to see what an individual SNP might be doing. Okay, this is just to motivate this a little bit. And I want to try to summarize for you how you can think, or how one can think about genomic data visualization approaches and what different kind of criteria you can consider when designing or building such approaches. So first of all, I want to build a little bit on the challenges that we have when you're visualizing genomic data. So first of all, the organization of the genome makes visualizing the genome tricky. We're generally dealing with multiple chromosomes in eukaryotes, so we're not even dealing with one sequence, we're dealing with many sequences. In fact, they don't have any particular order, right? I mean, we just order them 1, 2, 3, 4 based on the size. That goes back to many years ago when people just thought to sort the chromosomes by their size. It's stored, the information is stored as a linear sequence, but of course interactions are non-linear. Right? You can have promoter enhancer interactions, you can have a SNP affecting regulation of a gene that could be due to a change in how proteins bind to the sequence, etc. So these interactions can be sort of in the short range between a promoter of a gene or a transcription start site or transcription factor binding site. It can also be long-range interactions, right? This could be across chromosomes, essentially. And that is very important information when we're trying to interpret what is happening. We're also then dealing with the scales. I think I just demonstrated that and explained to you what these scales can be from the whole genome down to individual nucleotides. And finally, the types of data that we're dealing with is also very diverse, right? We might be interested in SNPs, which are individual nucleotides or individual positions in the genome. We might be looking at something mapped to the genome, such as epigenomic peaks for some methylation patterns or something. We could be interested in genomic rearrangements. There might be a interchromosomal translocation of a stretch of DNA that might be went from chromosome 1 to chromosome 17. And we would like to be able to see these kind of things as well. All right, that's not the end of the list, though. When we're thinking about interpretation, what's really clear, I mean, to everyone who does anything in biology, understands that all of these things are connected to each other, right? I mean, you can look at the sequence. The sequence will influence, for example, expression levels. Gene expression levels will influence information about metabolite levels. You have phenotypic information that you might have to integrate. And so you, depending on the question that you're trying to answer, you will end up with a different set of data types that you're trying to integrate. And that is not so much necessarily a specific visualization challenge, but that's a challenge when we think about building tools that enable people to interpret genomic data or omics data in general, because you need to be able to bring a lot of different data types, most likely in a lot of different file formats that are not standardized, into your tool. And it's not that there's a library out there that you can use to read all of them, right? You have to often pull from different sources and put them together into your software. Okay, now back to the patterns and the scales. So a genome is essentially a very large space with a sparse distribution of patterns generally across multiple scales. So sparse distribution, for example, SNPs, right? They are generally, I mean, we have a lot of them that we might be interested in, but they're very sparsely distributed along the genome. So a genome-wide overview, like looking at SNPs in that context, might give you a sense of maybe the distribution where they are, but it wouldn't tell you anything about whether that SNP would affect, say, expression or something else. So that's something to consider when thinking about visualization. We deal with large data sets in terms of the data size and the amount of terabytes or gigabytes of data we need to deal with. And when you're dealing with human data, you also, and this is really related to building software, you have to think about privacy, right? So you cannot just download a human genome and put it in your laptop and maybe build a little web server that allows other people to look at that human genome. So you need to have infrastructure that can ensure that you're at least HIPAA compliant, that not random people can just download genomes of people who are part of your study, for example. So that's something to think about as well with building genomic visualization tools. Okay, so that's an overview of some of the challenges you need to tackle when building visualizations for genomic data. I want to now tell you a little bit how to organize these kind of visualization tools and how you can think about them, and also how you can realize a way to really understand how few approaches we actually have to look at genomic data, but also maybe help you understand why we have so many different tools. So, as I already emphasized, we're only going to look at data that's visualized in the sequence context. So there has to be the genomic sequence somewhere in there. It doesn't have to be shown explicitly, but the data is mapped to that sequence and it has to be shown like that. Then I want to propose that we treat the genome as a coordinate system. That's not an unusual thing to do, but I will tell you in a second why it's important. And then when we analyze tools that are out there trying to understand what the space of visualization tools for genomic data is, we want to separate it concernedly. So first of all, you should think about what data types are, because data types are very different from how you visualize the data in the end. But you need to understand, we have, for example, SNPs are one thing, is one data type, but when you think about, say, structural rearrangements or genomic interactions, and you have 3D folding of DNA, that's a completely different data type. Right? And we want to look at how we can lay out the coordinate system that we're dealing with. Right? So you've already seen two examples of how the coordinate system can be laid out. So how is the genome laid out in circuits, and how is it laid out in a UCC genome browser IDB? Right? So we have either a circular layout, or we can have a linear layout. Then, of course, this is a big one. How can we visually encode the data that we're mapping to the genome? That's another question. And if you've played around with ggplot in the last couple of days, you've probably seen many different ways of how you can encode data. And of course, the same thing applies to genomic data as well. And we use pretty much the same approaches. In essence, we just have one axis, which is the genome axis, and then we have quantities, or we have just positions, etc., and we can all use that. And finally, and this is a question I'm not going to go into detail, it's like, what are people actually trying to do once they have a visualization of data, or before they even get to the visualization? That will depend on the data that they have and what they're trying to understand about their data. Okay. Let's look at the data. And I'm going to go through this fairly quickly. But just want to give you a high-level overview of this. When we look at data, and I'm introducing essentially a very simple taxonomy here, we can think about data as the data we're looking at essentially sets of features, genomic features, something that is mapped into the genome, the SNPs, the genes, interactions, etc. And they can be broken down into two classes. So there's a type of feature. So you can either have these block-like things. I mean, the first thing here, for example, are SNPs. They're sparsely distributed. The second thing here could be, for example, genes. And then we can have continuous data, such as, say, methylation patterns or epigenomic profiles. And we can also segment the genome, essentially, in continuous segments. So that's, for example, copy number data, and you have literally a value for every single part of the genome, but these are generally longer stretches of the genome. And then these are just the features themselves, and then they can be connected, right? You can either have no connections between your features, You could have connections within your genome. So this could be genome interactions, genomic interactions in 3D. It could be translocations in the genome, deletions and other things. Well, so they wouldn't be, sorry, deletions wouldn't be connected. So that's within the genome, but you can also think about connections between coordinate systems in the genome. For example, when you think about synteny, right, you map between two coordinate systems, right? You want to know how or where a particular block of DNA or genomic sequence that you may be found in human, or that maps into a mouse. And then you get a mapping between two coordinate systems. That's the data, and you probably recognize a lot of things in here already. The next part is about visualization. So once we have a data taxonomy, we can think about how to visualize this. And I want to emphasize there are three different pieces to that. So first we have the coordinate system itself. We have a couple of different attributes to that. Then we have the tracks, so where we're actually visualizing the data. That has, for example, encoding, how these tracks are aligned. And then we have the views. When you have a visualization tool, oftentimes it's just a single view. So the things that I showed you, for example, a circus plot is a single view. There's one view of the genome, there's just one representation of the coordinate system. It's at one scale, that's global, and then there's one focus point, and that's everything that we're looking at. And we'll get into that. So let's look into the coordinate system, how we can lay this out. So there are four parts to that, for the coordinate system. First of all, the layout. That's what we already talked about. There's a linear layout, for example. We can do the circular. We can do something space filling. I'm not going to go into that. But you can essentially take your sequence and you can fold it in a sort of fractal way and fill at the plane or your screen with the data. So you can then map, for example, colors on here. And you can also come up with a 3D layout of your genome sequence or genome coordinate system. So this would be three-dimensional. Partitions of the genome. So as I said, we have generally more than one sequence. We have multiple chromosomes. Either we lay them out sort of in a contiguous way, from left to right, where they're all connected, but we can also segregate them, the chromosomes, and we could lay them out like this. It's certainly possible. And if you haven't seen a tool, I probably won't show you one today, but that's a possibility as well. Then we can abstract the sequence, or the coordinate system. So either we have no abstraction where we literally see everything, there's nothing is filtered out, but you can also, for example, have a partial representation of the coordinate system when you abstract this out, for example, in introns. When you just want to see an exon sequence, like the coding sequence of a gene, you would not include the introns. You just remove those on your visualization. And you could then go even further and completely abstract the sequence where you say, okay, I don't even care about the size or what specific sequence there is. I just say, okay, there is a block, then there's another block, and there's a third block. So you could think about, here are 3 exons, just to say there's one transcript, one isoform that has 3 exons. And that's all you need to know in this case, right? Because in that case, you might just want to know which exons are part of the transcript. So that's abstraction. And finally, we can arrange these coordinate systems relative to each other. For the linear case, we can do them in parallel or in sequence, or we can also lay them out orthogonally, right? So this is 2 instances of the same coordinate system, or it could also be 2 instances. Microphone isn't really working, is it? These could be 2 instances of well, either the same genome or 2 different genomes. Right? When I do 2 different genomes, I can do synteny visualization in here, or in something like that. When I use the same genome and the same coordinate system, I could, for example, use this to show interactions in the genome between different positions in the genome. And we can also build different arrangements with circular layouts, so either they can again be parallel. This is essentially the approach, one approach that you could take with something like circus plots, or you could do this serial where you sort of put them next to each other. Okay. This is about the coordinate systems. And I hope you recognize in here many of the things that you see when you look at genomic data. And typically, actually, what you see is the first row. So you see a linear layout, contiguous partition, and no abstraction. That's essentially what you see in a genome browser. But occasionally, you'll come across other approaches. And I'm going to explain to you later on why some of them are used in certain settings. Okay. Track encoding. I don't want to go into details here, but you probably realize that there are many different ways how we can encode these different feature sets that I introduced in this data taxonomy when you visualize them. So the idea here of a track is essentially one data set that you can visualize or want to visualize. For example, here at the top in sub-panel A, these might be SNPs. The same thing in the second track here in A. These are just positions, and the color might indicate what it is. Or you could have blocks, and in this case, so the block indicates where the thing is, and the color might indicate a category, for example. You can also do this with continuous data. In B, here the colors for the blocks would not represent a category, but might reflect some quantity that is associated, say, gene expression levels. You can also do this with the height of the bar, or if you have continuous data, you can also plot sort of a wiggly line that represents a quantity. When you have two-dimensional, two-coordinate systems, you can visualize as a heat map, and this is what we'll talk about shortly, so I'm not going to go into much detail here, but this could show the strength of an interaction between any pair of positions in the genome. A similar thing can be done also in 1D, when you are in sort of along a linear track representation where you only have one representation of the genome, where basically you go from here, so this point basically is the interaction between these two positions. That would be the interaction between these two positions, so you can visualize that as well. And then, of course, you can build arbitrarily complex configurations by assembling multiple tracks, or even tracks and matrices, or in this case, maybe two-coordinate systems where we have a mapping between either two genomes, right? You could have maybe this is human, this is mouse, and you show these synteny blocks. But you can also actually use it to visualize interactions. Again, when we have the same genome, but maybe different positions in that genome, right? So I could show you the interactions between chromosome 1, that might be here, and chromosome 3. I just shift these genomes relative to each other. Okay, so this is what we can do in terms of encoding. So here we use, like, these bands that we're using here are the same as the blocks that essentially represent the interaction. Okay. View configurations. So as I pointed out, and if you just hold off for a little bit, there will be an example in a second. We have a typical standard genome browser. We have basically one view. So a view essentially defines what we call a scale. So it shows you some region of the genome that can be changed, of course, but it only can generally show you one scale. So what does this mean? When I have a genome browser, I can either look at a whole chromosome, or I can look at a gene, or anything in between, but I can never, but the standard genome browser, I can generally not look at both a genome-wide or a chromosome-wide overview, and at the same time also at a genomic or gene position, for example, or a SNP, right? So that means there's one window, and it has one scale, and it also has generally one focus. What does the focus mean? That means you can only choose one location for the window that you're interested in, either the whole chromosome, or you can zoom in into a particular gene, say TP53, you zoom into that, that's it. You cannot choose another gene or 3 more genes and to look at them as well in this configuration. Single view, single scale, single focus. But you can make this arbitrarily complex, right? So you can use multiple views, which is shown over here. We have multiple views. And when we have multiple views, we can either then put all of these views on the same scale and the same focus. That would be this thing here. So we might have 3 windows that essentially show us the same thing, but why would we use multiple views? Maybe because we want to have a circular layout and we want to have a linear layout. Just look at the data in different ways. But what gets more interesting, and that's really what I want to get to, when we have multiple views, we can also allow the viewer, the user of the tool, to look at the data at multiple scales. And why are multiple scales relevant? I've already tried to point it out earlier, right? So we might want an overview of where are the things we're interested in, where are all the SNPs. But then we want to be able to use that to select maybe a couple of SNPs and then look at those SNPs. So, of course, when we look at the SNP, we don't want to be on the same scale. We don't want to be on the whole chromosome overview. We want to be on, maybe a small region around that SNP. And that's what we could do with multiple scales. And now when we are already talking about multiple SNPs, this would mean we look at multiple positions in the genome, so multiple foci. And essentially with this little taxonomy of how views can be configured in genomic data visualization, you can describe any visualization tool for genomic data that's out there in terms of multiple views. You'll find that most of them actually fall into this category, which hopefully you understand and see already that that's quite limited. All right, some examples. The last part here is the task taxonomy, which I'm not going to go into today, how we actually try to interpret the data. Examples. So here's a tool that we will talk about shortly. It's called HighGlass. And I have annotated this tool for you. So what we're looking at is essentially a genome-wide interaction matrix. I want to ask you for a second, how many views do you see here? So there's one matrix, and you see 3 more. So we have a total of 4 different views in this example. The thing that you cannot really see, and so that's just indicated here, is the number of views. So we have multiple views. What you can't really tell from the back, we are looking at different scales. So this view here is a very large overview. So each block here represents a chromosome. This is this area that we selected. This area is enlarged here. So this little white square is this one here. And then you can keep going. And then this view has its own detail view, which is up here. And so this little white box in here is this view. So this is how this is configured. There are different scales. So we have a really good overview, and then some intermediate scales, and then here's sort of a detail view. At the same time, and this is something that is not, in this example, not really shown, we're always looking at the same position, essentially. There's one focus area. So we focus on this, and we just have that same window at different levels of resolution. But in general, this tool, HighGlass, you can also look at multiple different positions at the same time, and I'll demonstrate that. That's why we marked it here as being able to show multiple foci. Tracks here, so how are tracks aligned? So they're just stacked on top of each other. That's what's done here. So we have the gene tracks here, chromosome coordinates, and then here's some epigenomic signal. These are, I don't even know what that is. That's some calculated score we're looking at. So that's what this tool does in terms of tracks. The coordinate system here, again, is essentially we have a linear layout. It's the partitioning is contiguous, so the chromosomes are connected to each other. You can't see this in this example. You can only sort of guess it here because basically along the side we have chromosome after chromosome. That's what this thing means. The sequence is not abstracted, so we always see all sequence. There's no way to filter out subsets of the sequence. And then in terms of the arrangement of the coordinate systems, it essentially supports either just a linear thing, or it can do these orthogonal arrangements of coordinate systems. So we have that essentially here when you go up on the side. This is one axis, and that's the second axis. 2 coordinate systems in here. That's represented by this thing. And finally, the feature sets that it displays are essentially any genomic data, but specifically here with the interactions within the same sequence. So this tool cannot show you synteny information, at least not with this setup, but here it can actually visualize the connections. And in this case, the connections are shown as the colors in this heat map, the strength of the connection. All right, here's another example for the same problem. So this is a visualization tool called Rondo, that's also designed to visualize genomic interactions. It's a completely different approach. So here what we have is we have 2 separate chromosomes that we're looking at. Chromosome 16 over here, chromosome 10 over there in yellow. And what this visualization shows us is essentially what parts have some sort of level of interaction. And this is, now, here the colors just indicate basically what part of the sequence is somehow interacting with another part of the sequence. So there's a lot of interaction happening in this segment of the sequence. This is something that's very common. I mentioned this at the very beginning. We have short-range and long-range interaction. So DNA is generally interacting a lot in sort of the short range, like around a particular stretch of sequence, there will be a lot of interactions. That's why there are these blobs here. And in this case, they had to set a cutoff value for interaction levels, and that's why you basically see these segments here. The second thing you can see is there are, of course, also then these longer range interactions between different parts of the chromosome, but also interactions between the chromosomes. Now, the thing that you can see very easily here is the interactions between the chromosomes, in this case, because of their filtering approach, there seem to be a lot of them, but they're most likely, to be honest, not very relevant. There might be one thing, though, here between chromosome 10 and chromosome 16, this stronger band here, where we might have an interaction that's actually relevant. And on top of that, you can also see interactions with other parts of the genome that are not selected. So we only see chromosome 16 and chromosome 10. And here what we have are essentially colors for all the other chromosomes in the genome. And then these rainbows essentially indicate, well, you have an interaction with, say, chromosome 1 through probably 22 in this case, or 23. So, well, yeah, so we could guess that, but it's not particularly helpful in this case. But the point here is, again, we look at the same kind of data. Now we have a different layout of the coordinate system. It's just a circular layout. And this is the same in terms of the partitioning of the genome. There's no abstraction. And the tracks, again, are sort of stacked. There's not a lot of track information in this example. They can show a little bit more. But then this is the big difference. So there's one view, there's one scale, and there's one focus area. And so that's where the differences start. But beyond that, I mean, the visual encoding is, of course, different. We didn't include that in this example here. But beyond that, there's no difference. Okay. And now, as I pointed out, so visualizing and interpreting this kind of data is quite challenging. And I want to spend a little bit of time now to tell you more about visualizing genomic interactions. Yes?
I didn't quite understand track encoding exactly.
Yeah, let me show you this a different example. So this, for tracks, how the tracks are, here. How the tracks are aligned to each other. So we have basically only two options. So either you have them as a stack, or this indicates they could also be stacked or plotted on top of each other. So let me give you an example for that. When we go back to a genome browser, like this one. So here, each data set is it's a track, basically, in the genome browser. That's where this term is coming from. So each line here represents one track. And they're all stacked on top of each other, right, from top to bottom. That's what that indicates, and that's a very common approach. The reason why we do this is because a lot of interpretation genomic data is correlation. Like you just want to correlate something that you see in one track with something that might or might not be in another track. That's the reason why we align them in this way. The sequence position is always the same, right? So we have one coordinate axis, and everything is aligned to that coordinate axis, and we just stack them on top of each other. And in addition to, if you care about correlation, like is something in this location, is there something else in another thing, you can also do this by just plotting these things on top of each other. Do I have an example for this? Not in here specifically, but I think if I show you, yeah, for example here, we have, this is an example where the stacks are actually, where the tracks are plotted on top of each other. So you have two types of data here. There's one, this gray line at the top is just the read depth, how many reads map to this position. But then what they plot on top are these colored lines, which represent SNPs and how frequent they see certain variants. So a green line, I don't know what a green is, there's no example, but a completely, like the blue and the orange that you can see here is this position. It's essentially information about what variants are found in the reads that map to this position. So these are tracks that are on top of each other. So these are two different data types, right? One is sort of a quantitative measure, how many reads are there, and then the second thing that we map essentially information at that position. It's a categorical information essentially. Okay, good. Now let me move on and let's see. Sorry, I just got to get through that. Okay, so I want to talk about how DNA is organized in nucleus. Keep an eye on time, I think. So how is DNA organized in the nucleus? And I want to use a little example here that might be helpful and memorable. So if you think about the nucleus as a basketball, you can take a rope that goes all the way, or a string. It probably has to be a fairly thin string. It goes all the way from New York City to LA. And if you were to put that string into that basketball, that gives you a rough idea about how densely the genome is packed into the nucleus. And this is something that was first sort of used as an example in the 1980s. And I think this is quite helpful. I mean, other ways to think about how genome like stuff together and how dense it is like when you think about take a pot full of spaghetti and just take the spaghetti, put it in a bowl, and it's really, really tightly packed together and looks potentially very chaotic. But of course, this is not true for DNA in the nucleus, right? So we know that DNA is not just some random arrangement of sequences. In fact, there is a lot of structure in there. Here's an overview from a paper that introduced something called the 40-nucleome project, which is an NIH-funded project that is a large collaboration that studies essentially how the genome folds in 3D and how it changes under different conditions or over time. Right? So that's clear. We know it's not static because how genome folds, for example, influences gene expression levels. Right? So although if the genome was just a static structure, there wouldn't be much control over expression levels from that point of view. Of course, you can still deal with transcription factors, etc., but what transcription factors can actually access and the other transcriptional machinery depends highly on how densely the genome is packed in a particular region. I don't want to go into any details because it has very little to do with visualization. I just want to sort of point out there are different structures at different levels. So there's something called loops, which are sort of the smallest structures we care about. In this example here, so this is the loop itself. There are various proteins that form these loop complexes and enable DNA to fold like that. This is the lowest level. Then we see that, of course, coming together, and then we have things called topologically associated domains. Those are more densely packed regions, but regions that are generally closer to each other in the genome. And then, of course, we have sort of chromosomal structure. Right? Chromosomes, of course, as individual sequences and individual molecules by themselves are densely packed together, and, well, depending on what state we're in, but they are, of course, interacting a lot more with each other than with other chromosomes, and you will see this in a second. And then, of course, all these things that I already said can change over time. Okay, so why is it interesting? I just want to motivate this a little bit more. So what's the role of 3D structure? First of all, it really influences gene regulation. I already pointed it out. Structural variations and the impact of structural variants in the genome, in the genomic sequence itself, can be, in some cases, explained by how the genome folds. And of course, the 3D structure and how the genome folds is critical for cell division. Otherwise, you would not be able to properly copy any chromosomes and separate them into the daughter cells. So in gene regulation, right, we're dealing with, for example, things such as enhancers, right, where the spatial proximity is important to control gene expression. Right? So you need to bring the enhancer closer to the gene that it controls or influences. And in order for that to happen, you need to move the DNA around in space. We have, or we know that, for example, when we have chromatin clusters along the lamina, that can be an indication for gene silencing, that there are certain locations in the nucleus where genes will not be transcribed. And the last example here is, for example, when you have GWAS data and you try to understand what your non-coding variants are doing, or variants in non-coding regions, that is also something you can study when you understand the 3D structure of the genome, how these SNPs might be affecting other mechanisms or processes. In terms of structural variation, as I said, you can also, if you understand the structural arrangement, you can understand, for example, why if you understand the spatial arrangement, you can understand why certain structural variants occur or not occur, and how they might actually affect regulation and other things down the line. And cell division I already explained. Okay, let's go back to visualization. Before we can visualize anything, always critical, of course, that we understand the data that we're dealing with. So how is chromosomal conformation or 3D structure in the genome measured? I'll just give you a quick overview. In general, we use techniques called chromosome conformation capture. They're pretty straightforward, at least in theory, for people like me who don't have to do it themselves. So we cross-link chromatin. There's a digestion step where those parts of the sequence that are not cross-linked will be digested. Then you ligate them again, and then you can do a reverse cross-linking, and you can start sequencing these fragments. And you can do this in many different ways. So the first early techniques were called 3C, which just stands for chromosome conformation capture assay, which is, and this is important, which allows you only to study one locus against another locus. You can test whether there's interaction or not. But what we're using these days are techniques such as high-C that allow us to basically get genome-wide interaction patterns, so we can measure all positions in the genome against all other positions in the genome. A similar approach is GeoPet, which also is just from the experimental setup different, but in the end gives us similar data. Now, when I say all versus all, the resolution of these assays depends highly on the number of reads you can get out of your experiment, and of course there's costs associated with that. So typically, what we see for high-C data these days, it's about 1,000 base pairs against all of the 1,000 base pairs. So the blocks that I'll show you later, at which level we can show or look at interactions is 1,000 base pairs approximately. We can't go lower than that. You would have to sequence so much that it would be, A, probably not very informative, and also very, very expensive and hard to deal with the data set. So keep that in mind. We're not looking at base pair level interactions here. Okay, so high-C, that's the protocol. I was described in 2010, I think, or 2014, by Rao et al. That's sort of a slight advanced version of the high-C protocol. But again, it's pretty much the same idea. There's some cross-linking happening. You cut with the restriction enzymes, you fill it in, and you get these reads, and then this is what you want to sequence. And then there's a whole pipeline that allows them to map that back to the genome, and then you get essentially a matrix that tells you how strong the interactions are between any pair of 1,000 base pair bins in your genome in this example. So what can you see in terms of structure in here, if anything? So what do you think are these blocks along the diagonal here? Chromosomes, yes. So these are the chromosomes. This is a human genome. There are the chromosomes, so chromosome 1 all the way, and then that's the X chromosome down here. You also see some interactions, obviously, off the diagonal, but it's generally fairly weak overall. You also see sort of this dark line along the diagonal, and this is really, these local interactions are really the strongest ones and the ones that are observed most frequently. This is simply because the sequence is obviously connected. We'll zoom in a second and you'll see more structure, but you can see that within the chromosomes there's also a bit of structure. So here's sort of like this checkerboard pattern in there that you can see, and we will take a closer look in a second. But the first question here is, how big is this thing actually? So I already told you it's genome-wide, 1,000 base pair bins, so you can calculate the numbers very easily. It's a 3 million by 3 million matrix, which is pretty large, but how large is it? So, as I already pointed out, it's 1,000 base pair bins. These are the numbers. But what does that mean in reality? So, if I have 3 million by 3 million pixels, how big is that? So you could print it out in a standard high-resolution printer, and then you end up with something that's about 830 feet by 830 feet, or 250 meters by 250 meters, for those of you who think in metric units. But how big is that? So let's look at the Empire State Building. This is how big this matrix is. You can essentially print this out on a sheet of paper if you find one that's large enough, and wrap the Empire State Building in a pixel by pixel printout of your interaction matrix. So that is the space you're trying to visualize. And that's the space in which you're trying to find patterns and that you're trying to interpret interactions between different parts of the genome. Okay, so of course the question is, how can we address this? We could use a bigger screen, but clearly you're not going to find one that's big enough. And so let's think a little bit more about what we're dealing with. So here is an actual map of the Broad Institute in Candle Square in Cambridge. The Whitehead Institute is here as well, and a couple of other places. This is chromosome interaction or chromosome conformation matrix. You see we're in chromosome 8 in a region, we're looking at about, this is about 10 kb, so there are about 10 bins in here. So that's the highest resolution we can get. Okay, let's just look at what that means here. When we look at the Broad Institute and how do we navigate a space like an actual map, right? There are sort of some reference points in there. So we see the streets, etc. And once we start zooming out, for those of you who are familiar with Cambridge, you'll start recognizing neighborhoods. And MIT's down here. There's Charles River. Now we're seeing Boston as well. And if you keep an eye on this here, you see also some structures in here, right? So there are these blocks. You see these darker spots up here. And you see more patterns. There are some other dots in here. Now we're seeing Massachusetts and New Hampshire. So now we're sort of roughly at the point where we see a whole region in the United States. And here we happen to see a chromosome over here. And we can zoom out further and further. And it turns out, when we zoom out all the way, we realize that this matrix is pretty much the size of a world map. And now this is our job to navigate this, find places in there, and yeah, understand what's going on. So just as a, to give you something to think about how big the space relates that we're dealing with. And now, how are we going to address this, right? So how can we build visualizations? And you see one visualization of the data here, right? It's just a matrix. It's sort of the most direct visualization of this information. These interactions map to a color map up here, as you can see. And we see the chromosomes, that's great. And when we zoom in, we see other structures. But that is not very helpful, for example, when we try to see whether a particular change in the smallest structure would have an impact on the larger structure, because we can either see one or the other, right? If I want to know whether a SNP in a particular gene somehow, or a translocation in the genome somehow affects interactions, I would need to be able to see both the event itself that we're interested in and then the larger picture. And I'll give you examples of that. Okay, so what are some of the visualization challenges involved? I already went a little bit over that, so I'm going to move through this a little bit faster. So again, as a reminder, we need to see data at different scales, from the genome down to individual bins, so down to 1,000 base per bins. We want to be able, for example, to compare interactions across multiple conditions, right? That's another way how we learn. Like what I told you about the tracks, they're stacked on top of each other, and when you have tracks that represent different samples, say, epigenomic markers, say maybe some histone modifications, and you compare them in different cell lines, you can compare across, and you can start to interpret what's going on, right? So that's why we want to compare. We not only want to compare between conditions, but we also might want to compare features. Right? So we might want to compare, in this case, two of these TAD structures to each other. Like, do they look the same, do they look different? Both within a single matrix, but also maybe across multiple matrices. We want to, need a way to navigate this space, right? So unlike actual maps, we have a problem in genomes. There are not really a lot of landmarks. Right? When you look at a geographic map, you'll see continents, you know what that means. You can zoom in, you see countries maybe, or rivers and mountains and whatnot. Those are things you can use, at least if you have looked at a world map for long enough, you can use that to navigate the space a little bit. But in the genome, we don't really have that. One of the reasons for that is that the genome constantly changes, depending on what you're looking at. Although things such as the chromosome structures and the tags tend to be fairly stable. Okay, and then of course we want to do this in a web browser. We don't want any special custom software. We want to be able to just allow someone to go to a website and open it right there. Okay, a little bit of a brief review before we go into how we do this. To remind you, and I don't know if this was covered in the course, three main applications for data visualization, right? One application is to confirm a hypothesis, right? So that you can sort of say, I only believe it when I see it. That's a major application for data visualization. Another application of visualization is to present or communicate something, right? So sometimes you want to do that, like what I'm doing right now. And sometimes you just want to explore a data set where the task is to find questions, to come up with hypotheses, right? So those are the three main applications for data visualization. And of course, when you work with a data set, you're going back and forth between all of those things. That's indicated by these arrows, right? Sometimes you explore, then you maybe have a hypothesis, you try and confirm that. Sometimes you turn your exploratory visualization tool into something that you present, maybe by putting some screenshots into a slide deck to explain to someone what you've discovered. How, or what do we need to do in terms of confirmation in the context of genomic interaction data? Just as a general example, so this is sort of why we do want to confirm sometimes things. So has anyone seen this? Does anyone know what this is? So datasaurs does? It was covered. Okay, so don't need to explain that. What is the other example for that? Does anyone know the other thing that was there before the datasaurs doesn't? Did you cover that? Great. Ansible score 10. Okay, so that's those are some general confirmation problems. Here are questions that you want to confirm generally when you visualize high-C interaction data. first of all, does it look like a high-C interaction matrix? Do I see sort of these chromosome blocks along the diagonal, or does it look different? You know, sometimes you need to filter data. You can check that in an overview. You know, is the normalization, did that work correctly? Does it look the way it should look, at least at a high level? So those are confirmation questions, and here's an example of where it didn't work, right? So what you see here, so again, diagonals should be this way, but if you find things like this, these diagonals that go in the other direction, in this case, and not only diagonals that go in the other direction, but also these weird bands in there. This is a case where someone didn't properly remove multi-map reads, so there were a bunch of reads that mapped into different positions of the genome, and you end up with these weird patterns that you should not be seeing in your high-C interaction matrix. Okay, so that's an example for confirmation. Next, in terms of exploration, what do we want to do when we explore high-C interaction matrices? So, for example, right, we want to be able to see these interactions at different scales. And to do that, we need to support such things such as zooming and panning, right? Zooming in and out, panning, moving the map around. Pretty straightforward. But I'm pretty sure that after this next 45 minutes, you'll probably never forget anything about genomic scales and how important that is. So this will come up over and over. Comparison is another thing we want to do when we explore a data set, right? So we want to basically juxtapose two or more maps, or we want to maybe even overlay them, right? You could overlay two of these matrices, calculate some sort of ratio, and then visualize these ratios to see differences between maps. We also want to be able, as I said before, right? You want to be able to view and compare features within and across maps. And then, you know, to do that, we could maybe link two maps to each other so that when we navigate one map, that the other one follows. Or we might also want to extract out features. So there might be certain structures in there that, you know, there might be thousands of, or there will actually. In fact, there are thousands of these tabs or loops. And maybe we want to pull them all out and just put them all next to each other without looking at the rest of the genome, right? We want to maybe abstract a little bit. And finally, we also want to compare what we see in these matrices to other genomic data that generally is affected by how the genome folds, and vice versa. Okay, so those are tasks of exploration, and for presentation, fairly straightforward, right? We might want to label stuff in the visualization. We might want to be able to sort of track what was done and share that as well. And finally, we might want to save a state for a particular thing that we're currently looking at and share this with someone. These are things that are not specifically super visualization related, so I'm not going to go into much detail here later. But I want to tell you more about the confirmation exploration questions. Okay, so how can we visualize data like this? We've already seen things, so I'm not going to spend too much time on this. So first of all, just a reminder, we're dealing with a very large network, essentially. You could think about high-C interaction matrices or generally chromosome confirmation data as essentially a completely connected network where each bin represents a node, and you have an edge between every pair of bins, and then you have a weight for each of these interactions, right? And then you could say, okay, it's at a cutoff value, and then throw out all those edges that are below the cutoff, and then you end up with a subset. And that's an approach that some people take. But then when we lay out this network, one constraint is, you know, the nodes have to be in sequence order. That's very important. So, you know, if you lay out these nodes, and this would obviously not be a whole genome, you know, this order here has to be the genome sequence. Or in this case, when you take sort of a matrix visualization approach, they also have to be in the same order as these are in the genome. Otherwise, we wouldn't be able to interpret it. You know, this does not maintain the sequence order here, this force-directed layout, so we can't use that for visualization. But here are basically the two main approaches that people use. One is a layout like this, in a circular layout, for example, and then we draw arcs between nodes or bins, and you know, we can drop them if they are above the threshold or below the threshold, and we can also use color, for example, to show how strong these interactions are, and the same can be done with the matrix that you already saw. Okay, so we've talked about the heat map idea, we talked about a node link diagram, or an arc diagram in this case, and we can also do this in 3D, but with 3D visualization of genomic interactions is maybe useful for concepts or for some models, but you will not be able to interpret anything if you try to render an accurate representation of how the genome folds in 3D, because there wouldn't be any room, first of all, to see what's going on. You wouldn't be able to map any data on top of that either. So, you know, that's nice to look at, but not very useful for exploration or confirmation generally.
Excuse me.
Yep.
And so, what's your idea of trying to use reality to explore data to get into the third dimension?
What are my views on that?
Yeah.
Okay. So I think, again, you could use that to, illustrate this to someone. If you want to engage someone and try to give them a general sense of that, that's where it might be useful. I think still in virtual reality, you have the same issue, right? It's extremely densely packed. So to visualize it accurately, you would constantly, you would basically have occlusion because some DNA would be in front of what you might be looking at, and you have to cut it out. Then you have to zoom in really far, but then you don't have an overview, and the overview is very hard to interpret. So there might be a way to combine sort of these 2D approaches with matrices or these node link diagrams or arc diagrams with a 3D representation, but it is hard to do any sort of global stuff with 3D. Those are my views. Okay. Scale, as I talked about before, global interactions, local interactions, and then we might want to see individual features. All right, so there was a great review a couple of years ago, I think in 2017 by Bill Noble and one of the students. They reviewed visualization tools for the software, and I'm just going to quickly show you some examples here for visualizations that they described. So this tool is called JuiceBox. Generally what it does, it uses a heat map. You've seen that. What's really important here, and you might have heard about this before, is when you have a heat map like this, you cannot interpret this like actually quantitatively because color is a relative medium, and it will be strongly influenced by the surrounding colors. Right? You could not, in this example they were pointing out a specific feature, but here you have some red that is surrounded by light red, by pink. Because it's surrounded by pink, you perceive it as a darker red. If you have the same color somewhere else, but were the same color surrounded by darker red, you would not perceive it as dark. So that's a huge problem with heat maps in general. So you can sort of do a qualitative analysis of what you're looking at, but never try to read any values off anything. Here's the example. Should have known that this is here, and I could have explained it with this example. So here, it's 2 times the same color, this and this, with light red, dark red. It's not the case. These are the same color, but this one is surrounded by lighter colors, and this one is surrounded by darker colors, and therefore we perceive it differently. Right, so colors are relative medium. Keep that in mind. They're just, here's some examples where this is even more extreme, where you can basically make 2 colors that are actually the same look very different, or 2 colors that are different make them look the same. And this is, if you remember this dress example, nobody really knew what color it was until someone revealed it, I guess. So there are a lot of problems with interpreting colors. So keep that in mind. Never try to map the color that you see back to any quantity. Okay, so in this case, when we look at global interactions in JuiceBox in the same tool, it also uses this heat map approach. This is the tool called HighGlass that we looked at before. This it can also do these heat maps with the same caveats. It also applies, by the way, to grayscale, not only to colors. And we'll learn more about this tool shortly. This is another example for global interactions, visualized now as an arc diagram. So this is the WashU epigenome browser that can generate these kind of plots. Here, and that might be a bit misleading compared to what we looked at earlier, this is just chromosome X. This is not multiple chromosomes, so you always have to be careful when you look at these things, what is actually mapped to the circle. So these are interactions within chromosome X, the X chromosome, and obviously the limitations are very obvious here, right? One thing you can see is like there are not a lot of interactions between this position and this thing over here. There are a lot of interactions otherwise between multiple loci, but you can't tell really what's interacting with what because these bands are just overplotted so strongly. An issue with using lines or edges or bands to connect things is the number of, like the interaction strength would have to be encoded either by the line width or by color mapped to the line. And in both cases, you're really limited in terms of the range that you can display. So like line thickness is generally not a very good encoding for quantities. Again, it could be made used qualitatively. And one thing that's really interesting, I don't know if I have an example for that, but zooming into a circle is really complex to deal with. So, you can, of course, zoom into this region if you want to. You can make that larger. Let me just check if I have an example. No. You can zoom into this. Obviously, once you zoom in, always you lose context. So once you zoom into this region, you can no longer see the rest. But with circles in particular, the thing is, I zoom into this thing, then I have lines that go sideways. And if I zoom into, this thing here, the lines are going up because depending on which window you choose, your stuff will be upside down up there, right? When you zoom into that. But that's a bit of an issue with circular visualizations. That's not really a good solution to that. So just to explain again, these are global interactions with chromosome X. Here we have global interaction across the whole genome in this tool called Rondo. One thing I want to ask you here, I like to include this because it's an interesting approach, but it's also quite limited here. So when you look at this global overview, you see pretty much what you see in the heat map, right? You see primarily these within chromosome interactions, right? So everything is interacting in the chromosome that's indicated by these blobs here. And then there are some interactions between the chromosomes, but nothing, it's hard to see any clear pattern here. It just looks like everything is essentially connected to everything else. And if I would show you two of these things next to each other, I'd probably not be able to tell if anything is actually different. Here is your global interactions within a single chromosome. This is chromosome 11 in Rondo. Now you can see maybe that some of these bands are a little bit thicker, but again, this is all based on setting a cutoff value to remove interactions that you think are not real. But how do you determine what's not real, and what level should this cutoff be? That's a bit of a question. You also have local interactions. That's another issue that you need to think about. So here we're zoomed into a chromosome. Again, this is something called a 3D genome browser that can show you sort of small windows in the genome. So we're looking at chromosome 5 here in the region of a gene called ASAP1, and interactions of this position here and this position here. So this is what this arc indicates. This is the interaction strength between these two things. And then you have this sort of partial heat map representation as well. And this is again, so this position actually is indicated by this value here. I find this pretty hard to read because you always have to navigate this way to find what is interacting where. So for example, for this position and this position interact, you gotta go down here, go over, and then find sort of the intersection of these diagonal lines. So that's how you interpret this. And one problem here is like, although this is sort of like a strip that you can map in a genome browser, like all other genome browser tracks, the length of, or the range of the interaction is limited by how high this triangle is, right? If I want to show an interaction between, say, this position, or between like, so what we can see here, basically this position that's cut off here, basically this point, and this point, you can imagine if I double that, this triangle has to be basically down here, and so that will not scale very well. Okay, and then local interaction shown in an arc diagram. This is a small part of chromosome 8. That's also the genome browser. And so you see here, here's, first of all, you see sort of this, this is lower resolution data. You can see sort of the nature of these bins, right? So there are some bins in here, they're not explicit, but you can see this by where these connections are placed. And here's sort of a little bit of a scale, like these interactions are a bit stronger, and these are a little bit weaker here, but that's about it. So, okay, we're going to go a little bit further. So when we have matrices, how do we interact with them in Amplitude? And that's a little example. So when we do 2D maps in a tool such as HiGlass or others, what can we do here? So first of all, we can navigate like any other map-based viewer or matrix viewer, for example, by zooming in, right? So that's what I'm doing here. You see there's occasionally a remapping of the colors because now we're zooming into a region where the maximum value is different, and if you wouldn't do this, because the dynamic range of these interactions is so large, you would just basically see nothing if you wouldn't update the color mapping. The reason for that is that these interactions are, I don't know by how many orders of magnitude stronger than everything outside. So now we've zoomed in and we can see a little bit of structure. As I said earlier, we care about chromosomes, tags, and loops. So these darker spots in the corners here are loops. So there's basically interaction between these 2 points. That is a loop. Then these larger blocks that we see are TADs, topologically associated domains, that we're interested in. And we've already zoomed in too far in order to see an actual chromosome. Here we're currently looking at a window where we see a bunch of genes at the top. So this is chromosome 5, 54 million to 59. So there's about 5 million base pairs here at this point. So, and as you've seen, we can zoom and pan. That's a standard operation in these matrices. The second piece that I already pointed out, we want to correlate between other genomic data, and we do this in the form of 1D tracks. And when you do 1D tracks in a visualization tool such as HiGlass, standard operations. In this case, you might notice, unlike most other genome browsers, we have a continuous zooming and panning. So there's no reload or refresh of the view. So it's very smooth. And although you see some things jump around again because we need to adjust the scales as you change the size of the window, you can keep your focus in your mental model. Like this gap, for example, was always visible. When you zoom and then you click a button and then it takes a second for the next view to show up, that is generally problematic in terms of your mental model about what you're looking at. Now, one thing about navigating genomes, as I pointed out, is there are not a lot of landmarks. Although you could argue that genes, for example, are landmarks. And a lot of people are familiar with at least a set of genes that they work on or that they analyze. And that's something we can actually incorporate in a genome browser like this. So it's obvious that when you have a whole genome with some 25,000 genes, which is obviously only a very small fraction of the annotations in the genome, you cannot plot all the gene labels. But which labels should you plot? And so one approach that this HiGlass tool has taken is very simple. So we basically can rank genes based on how well known they are and how do we measure how well known a gene is, essentially by how often they're cited in the literature. So in this tool, you will notice when you zoom out all the way, so we are now at the whole genome level. There are all chromosomes at the top here. And a couple of genes are listed. And which genes are those? Oops, sorry, a little too fast. The genes that are listed are those that are highly cited in the literature or mentioned in the literature. So you see things like EGFR, and those are in fact mostly cancer genes. CDKN2A, which is a tumor suppressor gene, KRAS, and others are shown. So you can use that in the overview to maybe find where you want to go. Now, of course, you still can't show all of them, but in theory, you could also load your own list of ranked genes based on what you want to see in the overview versus what you don't want to see. Okay, so that's the 1D tracks. Now, another thing that I pointed out when we talk about exploration of this kind of data is we need to be able to link multiple views together in order to, for example, just support having an overview and a detail. How do we do this? In this tool, HiGlass, this is really straightforward because it's very flexible. Right now, we have a single view. Let's duplicate this view. So we have two of them. And now what we can do is we can resize that. In this case, I just put them next to each other. That's the point here. It's very flexible how you can build these visualizations in there. Put them right there, and then we have them almost the same size. Just that. And now this one is completely independent from this one. You see it can be moved independently. But what I want to do is I want to build an overview that helps me navigate and a detail view that allows me to see details. So in HiGlass, you can do this by saying I want to link the location between those two views. So I'm going to link the location of this one to this one. Now they're moving together. The zoom level is independent. Right? When I zoom out, so here's my overview on this side, and here's my detail view. You see the movement is very, very subtle because that's a high resolution view, and over there's my overview. Now I want to actually show or understand where I am, so I can also tell this view to show where this one is over here. So you see this little gray box there? This is this view over there. I can now move this around and use that to explore the map. So one problem that we just addressed is the scale issue. Right? You build an overview, you build a detail view, you connected the two, and now can use that to navigate. So we're looking at the same map still. Right? Both of them are the same matrix. There's no difference between the two. So this doesn't help us in comparing things to each other. So how can we do that? It's the same idea, right? We can actually now go in and say, we want to build a second detail view. The second detail view is now here at the bottom. First of all, you will notice what we're going to do now. We're going to replace this matrix with another matrix. we have a list of interaction matrices. I'm going to look at something called K562, which is a cancer cell line, which is now linked down here. But now this is again independent. These are linked, so because I want to compare these two detailed regions to each other. So I'm just going to basically link this view here at the bottom to the view at the top. Done. And now they're looking at the same location at the same zoom level. Okay. So now I can move them together, and I can now start comparing. Now, are the patterns the same in these two matrices because they're the same thing? Well, there's the same location but different data. There are no major differences here. So that's a fundamental approach. And I'm going to show you some examples of how this can be used. So this is from a paper called by Schwartz et al. And came out in Nature 2017, where we rebuilt their figures from my paper in this high-glass tool. And so what's the problem here? They were looking at mouse hepatocytes, and they had knocked down a so-called cohesin loading factor that is, well, they suspected is needed to properly fold the DNA. And so what we're going to see here on the left is the wild type. What we're going to see on the right is this loading factor is called nibble that has been knocked down. So here in the detail view, what you can see is this difference. You see here, there's no structure, hardly any structure along diagonal, while here you still see structure along diagonal. So the wild type does have these tags. In the knockdown, you don't see these tags anymore. So this is a nice example of why you want to compare these things to each other, and you can see differences right there. The other thing, and that is not visible in the detail view because it's zoomed too far in, is when we look here at the top, you see this sort of checkerboard style pattern up there. This is decompartmentalization inside in the folded DNA. That is not gone. That's still there. So these are two separate things apparently. I mean, that's the lesson learned from this experiment. That a decompartmentalization is not affected by the depletion of this nibble cohesin loading factor, as you can see at the top. In fact, this patterning seems to be even stronger in this other matrix on the left and on the right. When you look at here in the top, this pattern here, it's present here, but not quite as strong in the wild type. So this is one of the reasons why we want to have an overview and a detailed view, and why we want to have comparative views as well. And with this high-glass tool, it's very easy to construct whatever you need, whether it's just an overview detail, you want to compare two things to each other, or if you want to compare many things to each other, in fact, you can also set that up as well. So here's a second example that I just want to show you real quick, where we have a very detailed view of this region here. So we're looking at the wild type, the nibble depleted sample here at the top, and down here in the overview, that's also the nibble depletion. You see these weird dots, these pock marks along the chromosome here. They're not on the diagonal anywhere. So these are essentially just structures that nobody really knows what they mean or why that's there. But you can also see this sort of in the depleted example sample with these darker spots here. And I just go back. All right, so we can show this again. When you're looking at the detail, here's the wild type. There is a little bit of that, but it's hardly present. And then let me see. There's another one. You see a little bit of structure here. When you zoom out further, you see that's a little bit denser there. And yeah, so nobody could really explain that to us what that is, but that's something we found by exploring the matrix, because these things are generally not there, and they have no particular meaning here. Okay, so that's another example. Here's another example. So if you don't want to compare just two things to each other, you can compare 8 things to each other. So someone compared algorithms to identify these topologically associated domains, which, by the way, they can be nested inside each other. Here are 7 algorithms, and their results shown on top of one and the same matrix. In this case, it's this data here, and they did this genome-wide, of course. And you can see these boxes are the predictions by these callers. They're all very, very different. For example, here, the size of these tags is completely different from these. I mean, this thing calls gigantic things, this calls tiny things. It's very insightful, and you can compare these things with a tool like high-glass. Okay, I want to spend the remaining few minutes here on telling you more about another problem that we haven't tackled, and that's really hard to tackle with high-glass, and that's, what if I have 1,000 things I want to look at in a single matrix or across multiple matrices? The problem that we're dealing with in that case is the sparsity, right? Stuff is really far apart from each other in a genome. So how can we overcome that? Here's an example of what that might look like. So there is a matrix, and here these marks indicate what we are interested in. In this case, we're interested in these loops, right? These darker dots in the corners of these tabs. So here are a couple of examples highlighted. What if we want to just compare all of those in a whole genome? How do we do this? Right? There are a couple of thousand in there. Okay, and this is the second tool that I want to introduce to you, and it's called HiPiler. And you'll see in a second why it's called HiPiler. So we have many pattern instances, but they're sparsely distributed. Right, so how can we explore and compare them? So this is the tool that I'll introduce, and let me see if I can. Yeah. I'll fast forward a little bit because we've already talked a little bit about some of the challenges here. Reasons why we might want to do this is, right, so we have usually algorithms, many algorithms that call these kind of structures. Not only one, you've just seen an example for the tabs, there are also many loop callers. Right, sometimes you get very noisy results, and you just want to be able to compare them to each other depending which algorithm you use. Right? These questions we'll tackle, there are many different approaches that people have come up with, but our approach is the one that I want to tell you about. So, in order to look at many things at once in a sparse space, what we're going to do is basically cut this matrix into small pieces. You're going to get rid of all the stuff that you don't care about and just keep snippets, smaller, thumbnail-sized parts of the matrix and then compare them. So here's our matrix. These are the things we're interested in. They're just going to cut them out. And now once you cut them out, within each little block here, you still have the sequence information, but we're going to remove that from the global context. So now we're interested in some very local patterns in the genome. And once you have that, you can rearrange them in whatever way. You can put them on a grid. You can project them into using TSNE or PCA or something like that, and you can start exploring these snippets. And then you can also put them on piles on top of each other. So as I said, there are many thousands of those generally, and you don't necessarily want to look at thousands of those. You might want to group them based on their similarities. And then we have piles. Okay, so and then we might want to map them back to where they came from, right? For example, is this pile, those that are all very similar, are they all in the same region of the genome? Are they maybe all far away from the diagonal, or are they close to the diagonal? Are they part of specific regions, such as maybe transcriptionally active regions versus inactive regions, etc. Okay, HiPiler. So here's the tool itself. What we have on the left is an overview of a part of a chromosome 22 in this case, and here are loops that were identified in that chromosome. I want to briefly go through there. Actually, I'll skip that part. I don't need to explain everything. So here's the snippet view, or the pile view. There's an overview on the left. Okay, so what can we do here? This is the pattern we're interested in. This is a loop, this dark spot. In the center of these things, you can see other examples for that here. And one thing that you immediately see, all these snippets look different, and some of these loops are very sharp, and others are very fuzzy. Right? So this one is a very sharp one, this is a very fuzzy one, and then you have weird stuff in there like this, and the question is like, what is all this? So let's filter this a little bit. In order to do that, we will you can scroll through here, but you see there were some blank ones in there too. Let's just make these a little bit smaller. We get a bigger overview, and you see there's a lot of diversity in terms of these, in terms of the patterns that these algorithms identify. Okay, so what we're going to do, we're going to now arrange them in a 2D grid. First, we're going to sort them by the distance to the diagonal. You see those that are on the near the diagonal are all the ones up here, and they all have this weird diagonal pattern in there. That's just because they're close to it. And down here, we actually have empty ones, and you can also see the closer you are to the diagonal, the stronger and the sharper the peaks usually are, or these loops. And then it gets fuzzier, noisier. You can see that difference as well, right? These are pretty noisy. These are pretty smooth. And we can use that to organize them even more. So now we're going to get rid of all of those that actually have a diagonal pattern. There's this one pile. I'm just going to put them in the trash, and it's gone. So now we still have a couple of the ones I want to get rid of. I'm going to go through those quickly. So we're going to sort them now, or map them by noise and distance to our diagonal. And you'll see, when we make them a little bit larger, there are some that are, this is the noise level. They have zero noise, basically, because they're blank. Down there, and because they are in this view, they're sort of distorted a little bit, compress our visualization. Let's get rid of those. Another pile that we then throw in the trash. Okay, and now we have a space of loops that we might want to explore. And so what we're going to do now is we're going to aggregate them, regroup them based on how similar they are. So we have a projection right now mapped to distance diagonal versus noise. And what's going to happen next is that we're going to put them on a grid and then group them based on their position on the grid. So now we have piles in there, pretty neat. Here are piles. And the thing we can do now is we can actually go through a pile like sort of a stack of CD covers. I don't know if you're familiar with the cover flow interaction that Apple has, or still has, I think, in, to go through images and things like that. So we can change what the cover is, either the average across all snippets in there or the variance. And we can also sort of sift through there and see what's in this pile. Okay. So that's that. And now we can also understand context. Again, very important. We look at some very local patterns, but in order to interpret them, we might want to know where they are. And that's our context view over here, the global overview, or the overview itself. So now we have an overview, a detail view, that's a smaller area down here. And now we can actually see where these things are mapped. So this is over here, these are down there. And that's, by the way, the same. This is high glass integrated into this tool. And now I'm zooming into this one. There's my overview detail setup again as before. And now I can see exactly where these loops are down here on these patterns. And now I'm just looking basically here. This highlights only the ones that are currently visible in this visualization. The ones that are sort of faded out in the snippet view are those that are off screen, basically. Okay, once we have that, we can do a lot more. You know, we can inspect these piles in more detail. We could correlate other data with these snippets. We can do comparisons between data sets in this tool. We can do clustering, multidimensional clustering, and projecting these snippets, and so on and so forth. And then what we end up with in high glass, right, we have something that allows us to investigate both local and global interactions, because we can do overview detail, we can zoom and pan, and all that. But we can only look at a small number of features at a time. And here in high glass, you have a really strong focus on the local context, right? You look at a region, and then you have all the context for that region around there. But sort of to see global patterns in a tool like high glass, those have to be pretty large global patterns. They cannot be these loops. And that's what we can do in high glass. It allows us to investigate features across the whole map. It allows us to view hundreds or thousands of features at a time. But it has fairly weak support for the context itself. Okay, now I think I'm running a little bit short on time. I will see if I can just give you an idea. There's something in between, right? Maybe you've guessed that you could also imagine to combine those two things. In fact, it's something we've been working on. There's an approach that we call scalable insets, which is shown here, make it a little bit larger, where we integrate this idea of showing the snippets in the context of a global heat map. And you can, you know, sort of basically get a preview of what would I see, what features would I be able to see if I zoomed into that region. So this is what's here. And if you zoom in here, you have these kind of patterns there, and they're piled on top of each other. And that's a lot of fun in these matrices if you want to do that. But what's even more fun, I'll show you that in a second, is this. You can use it for high C maps. You can also use this to, for example, explore gigapixel images. So when you have really large photos, same problem, right? You can't zoom in and at the same time get the context. So generally you see one thing at a time, and this is a huge image of Rio de Janeiro, and you know, you have a lot of detail there, but you can only see it when you zoom in, and so you can take this scalable instance approach and map it on top. You can do the same thing with maps, geographic maps. This is an overview of ski areas. You know, you might want to figure out what are all the ski areas like in New Hampshire or in New England or overall here. Right, so we have these little markers, but so if you want to know, you know, how many runs are there, etc., you can't see it until you zoom in, because there is information about what these ski areas look like. But you can now, with the scalable instance approach, you can actually hover over this region, and you can see all of the different ski areas in there. That's scalable instance. I want to skip the high glass architecture. We spent quite a bit of time on this project to figure out how to build the architecture and how to actually support these kind of smooth animations and all that. I just want to, that's a different question. I want to point out there are lots of open challenges still we need to address. For example, one thing about genome interactions is they're actually not pairwise, right? So what I showed you are all pairwise interactions, but there are many scenarios where more than two loci come together. How do you visualize that? The heat map is definitely not the way to do it, because that can only show you pairs. But maybe you need to slice it in different ways. That's quite problematic. And single cell data is a different story. In that case, you would just have a lot of samples, most likely, a lot of different genomes to show how they fold. And finally, visualization of temporal dynamics. How do you do that? Like, how does this matrix change over time? Under different conditions, that's quite a question that nobody has really addressed yet. I want to leave you with some lessons learned here about that 3D visualization. Right? So this HiGlass tool essentially is a genome browser. Hopefully, if you think back to what I showed you at the beginning about how genomic visualization tools can be organized, HiGlass does have all of that, the tracks, the different arrangements of genomic axes and coordinate systems. One of the things that are important that I think we don't need to cover in this setting, when you build these kind of tools, it's really important that you can actually load the data files for workflows that are typically used to process this data. This last question here, I apologize, this was from a different presentation about, what kind of infrastructure do you use? You can build these kind of visualization tools, these web browser-based visualization tools without a server backend, where you have no need to run anything for anyone. Everyone just gets the data, has a little bit of JavaScript code running in the browser, but then you don't need to maintain anything, but it has also certain limitations, actually a lot of limitations, and therefore we decided to build HiGlass with an actual server in the backend that provides information. Okay, and these are the two guys who build these tools, Peter Kopedjev, who used to be a postdoc with me, and Fritz Lekschus, who's a PhD student at the School of Engineering and Applied Sciences. With that, thank you for your attention, and I'll still take a few short questions. Thank you. Yeah, thanks.
This is really cool, actually. So I was wondering that when you have a project of developing some sort of genome viewer, for example, and then you have all those principles, what you want to incorporate in the viewer. So in terms of actual implementation of that tool, can you please give an idea of what pipeline you guys follow, like in terms of what software you use, or whether you integrate some GUIs with some software, or you write your own code? How do you develop the pipeline of starting a project until the end product?
Okay, so that's an excellent question. The most important part, actually, in for example, this high pilot project, was to understand the problem space, the problem domain. So we started by talking to people who analyze this kind of IC data on a regular basis and who are experts in processing this, analyzing this kind of data, and tried to learn from them. What do they actually need to do with it? Right? Because we are bioinformatics, but we're not experts on IC data. So with that, we were able to come up with a set of tasks, visualization tasks that we need to support, and some requirements for the software itself. And so one question, we want to compare groups to each other, and many different ways how to solve that, obviously. But we also needed to understand how many loops are there and what kind of software do we need to have to support that. So we did that, and then we knew that we wanted to base that high pilot tool on the existing infrastructure we had built for HiGlass. And HiGlass is fairly modular at this point, so we were able to integrate this easily. But we had to build a lot of software around this, and for that we're generally using standard web-based development tools. And there's compilers and HiGlass, both React-based tools. Use D3 fairly often to implement things. HiGlass itself is actually not as much D3 as you might think. It's mostly actually a gaming library that renders the matrices, because that's something that we use hardware acceleration for that, because you have to draw a lot of small boxes, and that takes actually quite a long time to try to do this with something like D3, which creates essentially an entry for every element in the document that you're dealing with, essentially the HTML document. It's in browser memory, but it takes a long time to modify these structures. So we're just using hardware accelerated rendering to render the matrix, and stuff on top of that is still rendered in D3. We do that. In the case of HiPilot, we didn't have to develop a server because there was a server already for HiGlass. The HiGlass server, for example, is written in Python. Really straightforward Django web application, really. And what it does primarily is provide tiles. So you request a tile, say, okay, for this window at this zoom level, give me the data that's there, and then the server will send it back. Beyond that, I think it's sort of pretty standard web application development with, I think, the awareness that most standard web, like graphic user interface components actually don't fit so well into these visualization tools. We do a lot of custom development for that. For these controls, for example, if I just go back to this example here. You know, what you saw down here. Oh, that was too far. The one issue that you have is like, you know, you want to have like this controller down here. You know, how do you? Well, so this is just a simple thing. Sorry, get rid of these. Now we're going to modify this. The noise that was also straightforward. Sorry, I want to show you this thing here at the bottom. This one, for example, right? So this is not a component you can just get from somewhere. So that is custom made. All the buttons and stuff are implemented by us. So that, yeah, that's necessary sometimes in visualization tools because you know, when you use standard components, they will be designed for certain uses that are generally simpler than what we do.
So these additional things that you add, are those written in some JavaScript?
Yeah, that's all written in JavaScript, CSS, or we're using SAS most of the time. And yeah, it's pretty, nothing super fancy, but generally it takes some time to acquire the skill level that you need to. I mean, this is a very advanced project created by someone who spent a lot of time developing web-based visualization tools and also has a fairly good design sense. So not everything will look as great as this.
So, and also, do you guys incorporate the plot libraries like in MATLAB, Matplotlib, or from Python, Plotly?
No, so all the rendering is done in JavaScript in the browser. So we're not using any Python-based rendering here at all. It's all D3. You know, React is not really rendering, but for the software or the application, we use React. I'm trying to remember what else we're using.